Okay, so in the last video we talked about how the complementary strand synthesis of DNA in our genomes is performed by DNA polymerase. And we talked about the chemical mechanisms which these polymerases employ, in which they need to activate a 3' hydroxyl to facilitate nucleophilic attack on the 5' phosphate of a DNTP, a nucleoside triphosphate. We talked about how this means that synthesis by these polymerases is always going to proceed in the 5' to 3' direction. So you're never going to get a nucleotide added to the 5' end. It's always going to be to the 3' end. Make sure you know that. We talked about how polymerases catalyze this reaction by harnessing metals, these magnesium ions that present lots of positive charge. We also talked about how it's really important to know that DNA polymerase requires a short stretch of duplex DNA between the template and synthesis strand in order to be able to synthesize off of that. And that little short piece of DNA that's required to be annealed to the template is called a primer. And this double-stranded DNA stretch here, mediated by the primer, is going to be important for helping facilitate the addition of the correct Watson-Crick-Franklin base pairs to the growing helix, because the already existing double-stranded DNA structure is going to make it more favorable to add the specific Watson-Crick base pairs that are going to stack really nicely with that existing structure. And so those things working together make polymerase go and have high accuracy. But of course, mistakes can happen. And what happens if DNA polymerase makes a mistake? Well, the consequences of that would be that the sequence that contains, the information to make proteins and things like that is been mutated. And that can mean that we're not going to be making the things that we're supposed to make, and that can have big consequences for how life goes on. And so in this video, we're going to talk about how DNA polymerase deals with the mistakes that it makes. And that's through a proofreading activity that the polymerase actually has. But before we get started on that, we first have to think about what types of mistakes polymerase might make and what it's going to need to fix. And it turns out right from the get-go, polymerase is pretty high fidelity. So it makes a mistake about 1 every 10 to the 4th or 10 to the 6th bases, depending on the different species that you look at. And that seems pretty good, 1 in 100,000. That's a pretty good error rate. But if you think about how many bases your genome might be made of, so a human genome contains over 1 billion base pairs of sequence, and so if you're making an error every 1 in 100,000 nucleotides with a genome that's that big, 1 in a billion, 1 billion, you're going to end up making a lot of mistakes every round of replication if this is your error rate. And so we're going to have to find some ways to correct this. And so what kind of mistakes are out there? Well, one of the mistakes that happens that causes an incorrect nucleotide to be added to the growing chain comes from the fact that while nucleotides, and in particular the nucleobases that we've learned, predominantly exist in the structures that we've drawn out and that we meticulously practice learning to draw, occasionally a nucleobase will undergo a process called tautomerization, in which it switches into a rare form. And the key thing to know about these tautomeric forms is that the position of the H-bond donors and acceptors in these tautomeric forms is different. And so to show you what I'm talking about here, if we look at this guanine in the classic way in which you've learned to draw it, we've got this carbonyl group here that's an acceptor. We've got this NH, which is functioning as a donor, and we've got this amine here, which is a donor. Now, if we were to tautomerize this form, what that means is that we're going to switch this ketone group here to an enol group. And so basically what you can imagine is that you're going to undergo a proton shift where this hydrogen is going to be transferred to here, and then the resonance, you'll create the double bonding structure that matches that. So over here, what you can imagine is this H gets transferred to here, so then this double bond is going to come down here, and you get what's called the enol form of your nucleobase. And what you'll notice is that now we have an OH where there was a carbonyl, and so we've converted an acceptor into a donor. And similarly, where we had an NH, we now have a lone pair here. And so we've converted this donor into an acceptor, okay? So this process of tautomerization here has actually swapped the distribution of acceptors and donors on our nucleobase. And of course, this is very bad because the pattern of donors and acceptors on our nucleobases is the reason that we have the specific Watson-Crick-Franklin base pairing interactions that we're familiar with, right? So, for example, when we have thymine in its common form, we have an acceptor and a donor here that interact with a donor and acceptor in adenine. However, if we were to look at the, where's the rare form here? Well, let me choose a different one because these pictures are not symmetric. I apologize for that. So if we were looking at cytosine, for example, in its common form, we have donor, acceptor, and acceptor on the cytosine here. And if we undergo this tautomeric shift, what we now have is an acceptor and a donor in these positions instead. And so whereas normally the pattern of donor and acceptors enables cytosine to form a beautiful base pair with guanine, now by exchanging, making this an acceptor and this a donor, cytosine in this rare tautomeric form can base pair with adenine. And so these base pairing arrangements between rare tautomeric forms can lead to misincorporation into the growing DNA strand. So these are bad. That's one way that mistakes are made. Now, in addition to these tautomeric mistakes, there's other kinds of errors that can happen during other contributions to mispairing that occurs during DNA replication. So there can be, for example, non-tautomeric forms of bases. So for example, if your lone pair of adenine, for example, here were protonated, then this can enable it to form some alternative hydrogen bonding interactions with non-Watson-Crick-Franklin base pair nucleotides like cytosine. Or even in some cases, if you just rotate the bases around in strange conformations, you can come up with some hydrogen bonding patterns, for example, between thymine and guanine, like you see here, which are acceptable. And these sort of mispairings that are acceptable enough to sometimes make their way into the helix are called wobble pairs. And these, of course, are no good because you don't want to put a G when there's a T, you want to put an A. Okay, so these are the mistakes that kind of happen. So how can polymerase deal with them? Well, the nice thing about DNA polymerase is that it has in addition to the polymerase site, which is where nucleotides are added, polymerase also has a 3' to 5' exonuclease active site on it. And so that exonuclease site, it can perform nucleotide removal from the end of a DNA strand. And in particular, the function of this exonuclease domain in the context of DNA polymerase is going to be to remove mismatched nucleotides which are introduced during DNA synthesis. And so in principle, what we're saying is if you added an incorrect nucleotide, you could pivot your chain into this exonuclease site and then snip off the wrong, the mistake that you'd made. And so how is the incorrect base actually detected? Well, you can imagine that while there might have been enough hydrogen bonding or Watson-Crick or hydrogen bonding interactions to be able to add that nucleotide in, once that nucleotide is attached to the growing chain, it's not going to be able to perform the beautiful sort of base stacking interactions that we talked about in the context of the overall double-strand DNA structure. And so that means that the ends, the 3' end of our growing chain is going to be somewhat destabilized or more flexible than when a correct nucleotide would have been added. And so this conformational flexibility allows for the 3' end to sort of flop around and find its way into that exonuclease site on the polymerase that we were talking about. And once it's in the 3', once the 3' end has actually entered the exonuclease site, now, because it's there, the terminal nucleotide can be removed. And once it's removed, what this means is the polymerase is going to have a second chance to try to add the correct nucleotide instead. And so this process of following addition of incorrect nucleotides, allowing for the 3' end to sort of sample about and try to access the 3' exonuclease site, that gives an opportunity to fix the mistake and start over. And this proofreading and error correction mechanism that I'm describing to you here actually improves the fidelity of DNA replication by 2, sometimes even 3 orders of magnitude. And so if you think about that starting number where we were saying it's between 10 to the 4th, 10 to the 6th bases before it makes one mistake. Now we're getting up into 10 to the 7th, 10 to the 8th, even 10 to the 9th error rates. So that's going to be a substantial improvement over things here. And to kind of show you what this looks like, there's this video here of a polymerase, and here it has the strands, and suppose an erroneous nucleotide got added. So what you're going to see is that the structure is able to undergo a conformational change, which places the erroneous nucleotide in this exonuclease site up here, and that's going to allow for that to be cleaved off, and that gives us now a second chance to add the correct nucleotide. And so this process happens basically whenever an erroneous nucleotide is introduced. There's a chance for the chain to be passed to this exonuclease site to be trimmed back. Okay. So to sum up what we learned now about the proofreading part of DNA polymerase, we observed that mistakes happen. You know, the polymerase is really good, but it's making about one mistake every 10 to the 4th or 10 to the 6th nucleotide additions. And the types of mistakes that it's going to make arise from the tautomeric forms of nucleobases or wattle pairs that can lead essentially to the addition of a non-complementary nucleotide to the growing strand. And these mistakes, because they're disrupting the helical structure at the end of our molecule, those instabilities can allow for the 3' end to get passed over into a 3' to 5' exonuclease site on the polymerase. And once it's in that site, the erroneous base can be removed, and we get a second chance to add the correct base instead. And so that's pretty much the gist of the proofreading polymerase activity. And we now know how DNA polymerase can make DNA, and we know how it can proofread that DNA to make sure it's doing a good job. But we still have to figure out how you're going to use that polymerase activity in the broader context of trying to copy the entire genome. And it turns out it's going to be a little bit more complicated to just to take polymerase activity and make it successfully copy an entire genome. And it's going to require some helper activities to get the job done. And we're going to talk about all of those activities and how genome replication is achieved in the next video.